Thank you. So my name is Mihai Avram. I will be presenting on behalf of Gabriel Castrillon, the guy with the mustache you can see here, and Valentin Riedel. In this project, they tried to quantify the energetic cost of the brain in humans in vivo. I have no disclosures, maybe just to say that I was not involved in this project. Now, the idea behind this project, or the motivation, was that for its size, the brain consumes enormous amounts of energy. And earlier studies have shown that this energetic demand is linked to chemical signaling. What you can see here on this slide is that around 75% of this energy is actually spent at the level of the synapse. However, it is unknown how this maps across the brain. So how does this link, for instance, with the connectome? This is what their study is all about. It was a multimodal imaging study with simultaneous FDG PET and arterial blood sampling and resting state fMRI. From the PET, a metric of cerebral metabolic rate of glucose was derived, and you can see map in one individual subject here. From the resting state fMRI, the degree of functional connectivity was computed, and you can see a map in the same individual below. Now, in this individual, the two metrics are related, so there's a positive linear relationship between them. This was done voxel-wise, so the dots you see there are voxels in the gray matter. So we see that the degree of functional connectivity explains a bit less than a third of the variance in the CMR glucose. Now, if the degree of functional connectivity is actually regressed out of the CMR glucose, they observed that some voxels had higher or lower values than predicted by the model, indicating that some regions in the brain deviate concerning this energetic demand. What you see on the left is in one subject, what you see on the right is across all subjects, so this relationship was seen in all subjects. Now, if we plot these values on the brain, an interesting pattern emerges. So this is here across participants. You can see that regions of the brain that have high energetic cost of signaling mostly cover transmodal cortices, whereas regions with a lower energetic demand are more in motor or primary sensory regions. What you see on the right side is a region-wise distribution. The dots reflect participants, and the regions are color-coded. What we see again is that some regions fall below the line, so have lower energetic cost, while others have high energetic costs. The ones that have color are regions that had higher or lower costs in over 95% of the participants, so a very robust result. And again, in line with what we have seen before, sensory motor regions have low energetic costs, while associative regions have higher energetic costs. So the next question was if there might be some genetic underpinning to this deviation in energetic cost. So to do this, they correlated this energetic signaling map with gene expression maps derived from the Allen Human Brain Atlas. I think over 8,000 genes were investigated. 617 of these were significant. So following on this analysis, they did the gene enrichment analysis in which they tried to relate the function of these 617 genes. And what they found out was that 95% of the genes were related to signal transduction. seen here in green, and what you see here in pink is that 40% of these genes were related to metabotropic signaling, so G protein coupled receptors, indicating a relevance for slow-acting neuromodulators like dopamine, serotonin, norepinephrine. However, these are transcriptomic, so in the next analysis, they used PET maps, which were made available a couple of years ago by Hansen and colleagues. 19 such maps are available, covering both receptors as well as transporters. They investigated the link between these maps and the signaling cost map. A partial squares analysis was used, and the first latent variable across all these PET maps explained 86% of the variance in the signaling cost map. Now, in the next step, they evaluated these PET maps individually, and what they saw was that 5 of the PET receptor maps were significantly related to the signaling cost map. 4 of these were related to metabotropic signaling. Now, in the next step, they thought to investigate whether these regions may have been relevant to our human evolution. So they related the signaling PET map with morphometric atlas describing homologous regions from the chimp to man. And what they found out was that regions that were more novel in our, or that are more novel in our evolution and that have expanded more are coupled also with a higher energetic cost. Now, as these regions also cover or are highly relevant to cognitive functions, they wanted to test whether, or they wanted to evaluate the energetic demands of distinct cognitive processes. For this, they used another independent data set, the neurosynth. The neurosynth contains information from thousands of task fMRI studies that have linked specific functions to a specific region in the brain. 25 functions were evaluated, and what they found was that such high cognitive functions as reading and memory are actually related to these regions in the brain that have a higher density of neuromodulators. Now, as these functions are also known to be altered in psychiatric disorders, they evaluated whether their signaling cost map might be relevant for such disorders. To this end, they correlated the signaling cost map with distinct symptom axes. These were derived from a study by G and colleagues. It was published a couple of years ago, in which behaviorally assessed symptoms were actually grouped. I mean, they used the PCA analysis and grouped into components. 5 such components are available. Of these, only the component pertaining to psychotic symptoms was significantly related to the signaling cost map. Now, this finding gave them incentive to search further, and they evaluated then the relationship between the signaling cost map with gene expression maps of distinct psychiatric disorders. These were all derived from larger GWAS studies. You can see here on the slide schizophrenia, bipolar, and depression. ADHD was also evaluated, and autism spectrum disorders. However, the signaling cost map was only related to psychotic disorders, indicating relevant link here. So in conclusion, high cost regions are evolutionarily novel and involved in cognitive processing. The highest cost was found for slow signaling, so for metabotropic signaling, and high cost regions map along psychosis spectrum disorders. I would like to thank you for the attention on behalf of the team in Erlangen and Munich.